Active transport. There are many times when a cell needs to move substances against their concentration gradient, from low concentration to high concentration. Since this doesn't occur naturally, the cell must expend energy, usually in the form of ATP, to make this happen. The transport proteins that move substances against their gradients are carrier proteins. One reason cells need to maintain higher concentrations of certain substances within the cell than outside the cell is to maintain a voltage across their plasma membrane. This voltage, specifically referred to as membrane potential, is electrical potential energy, thus acting as a battery. Cells maintain a membrane potential of -50 to -200 millivolts, the negative meaning that the inside of the cell is negatively charged relative to the outside of the cell, due in large part to the presence of many negatively charged phosphate groups. Two forces drive the diffusion of ions across a membrane, an electrical force and a chemical force, which is essentially the concentration gradient. These two forces together constitute an electrochemical gradient. The effect of membrane potential is that it favors the movement of cations into the cell, since the inside of the cell is negatively charged and opposite charges attract. Likewise, the positive charge on the outside of the cell favors the movement of anions out of the cell. A transport protein that generates voltage across a membrane is called an electrogenic pump. The major electrogenic pump in animals is the sodium-potassium pump. The main electrogenic pump of plants, fungi, and bacteria is a proton pump, which creates a proton gradient. Because of its importance in animals, we're going to take a closer look at the sodium-potassium pump. Many cells expend a great deal of energy on their sodium-potassium pumps to maintain membrane potential. In neurons, for example, which are the cells in your brain and hopefully quite active, about 2/3 of their energy is put towards this pump. The sodium-potassium pump is a carrier protein that can bind both sodium and potassium depending on its conformation. In the first step, cytoplasmic sodium ions bind to the sodium-potassium pump, which has a high affinity for sodium in this shape. The binding of sodium ions stimulates ATP to phosphorylate the carrier protein. Simply put, ATP donates one of its phosphate groups to the protein. Phosphorylation causes the protein to change shape, decreasing its affinity for sodium, which is expelled to the outside of the cell. The new shape has a high affinity for potassium ions, which bind on the extracellular side and trigger the release of the phosphate group. The loss of the phosphate group restores the protein's original shape, which has a lower affinity for potassium. The potassium is released, so the affinity for sodium is high again, and the cycle repeats. At this point, you're probably wondering how this maintains membrane potential, since both ions being transported are cations. However, notice that for every 3 sodium ions that leave the cell, only 2 potassium ions enter the cell. Thus, a net negative charge of 1 is gained for every cycle of the pump. So, what is the point of all this? When a cell needs to do work, it can tap into the potential energy that is stored by the high concentration of sodium ions outside the cell membrane in the extracellular space. So let's return to those hardworking neurons in your brain that need a lot of energy in the form of glucose. They can open up a sodium ion channel, and sodium ions will move down their concentration gradient and return to the interior of the cell. This is favored not just by the concentration gradient, but also by the electrical gradient as well, which favors the movement of positively charged ions into the cell. The energy released from their movement down their concentration gradient can be used to move another substance against its gradient, glucose, for example, which your neurons can break down for more energy. Another type of pump that exists in many organisms is a proton pump, which creates a proton gradient. Proton gradients have many uses, some of which will be discussed in detail later. In a mechanism known as cotransport, a substance that has been pumped across a membrane, oftentimes protons, can do work as they diffuse back across the membrane. For example, plants use cotransport to load their cells with amino acids, sugars, and other nutrients. So in the diagram below, you can see a sucrose transporter. So plants expend energy in the form of ATP to create a proton gradient. Then the energy of the proton gradient is used to transport sucrose. As the protons go down their concentration gradient, the energy is used to transport sucrose against its concentration gradient. If you have any questions about any of this content, please don't hesitate to contact me.